In the heart of the Middle East, a unique laboratory harvests one of nature's most potent medicines, scorpion venom. This is the Scorpion Venom Research and Manufacturing Center, a fusion of biotechnology and desert ecology.
Hundreds of scorpions are bred and studied here, each species producing unique venom compounds.
Each scorpion is tagged, measured, and logged before entering the venom extraction phase.
We monitor growth and exoskeleton shedding to determine the optimal extraction cycle.
Under ultraviolet light, their bodies glow chromeware, a natural defense that helps researchers identify healthy specimens.
Each scorpion's venom yield, health, and feeding are digitally tracked for traceability.
Automation helps sustain thousands of scorpions with minimal disturbance.
Selective breeding ensures venom consistency and potency for pharmaceutical use.
Even logistics here follow biosafety protocols to maintain venom purity.
All collection vials are sterilized and coated before each extraction cycle.
Precision tools are essential. Venom extraction must stimulate, but never harm the black mamba.
We use microelectrical stimulation to release tiny droplets of venom safely.
Each extraction session is traceable. No sample moves without full digital approval. We collect, label, and immediately store each venom sample for cold transfer.
Automation reduces human contact, protecting both worker and sample.
We sanitize between every specimen to avoid cross-protein contamination.
Constant training maintains ethical standards and technical precision.
Venom is frozen within seconds to preserve its bioactivity.
Thermal sensors monitor colony activity without intrusion, minimizing stress.
Every terrarium's humidity and diet schedule are pre-programmed and self-adjusting.
With extraction complete, the next stage begins: purification and medical refinement. Once collected, scorpion venom enters a high-security laboratory for purification and analysis.
After collection, each scorpion is returned unharmed to its enclosure.
Each vial is traced through digital ID to ensure full biosecurity compliance.
The venom is gently thawed under strict temperature curves to preserve its delicate proteins. Filtration removes impurities fluid, leaving behind pure scorpion venom ready for scientific study.
These peaks represent different toxin families, each with distinct pharmaceutical potential.
Every droplet is analyzed to identify medically active peptides within the venom.
Mass spectrometry allows us to define each compound and structure at the atomic level.
By sequencing multiple scorpion species, we can link genetics to venom potency.
Digital simulations predict how venom proteins could bind to human receptors, paving the way for new drugs.
DNA amplification helps isolate the genes responsible for venom production.
Researchers replicate venom peptides using cultured cells, reducing dependence on live scorpions.
Maintaining balance between growth rate and peptide expression is critical.
Automation allows thousands of venom samples to be tested for medical potential.
Each test checks for pain modulation, antibacterial, or anti-cancer properties.
The Venom's chemical code reveals potential to treat autoimmune disease, cancers, and neural disorders.
We grow microcrystals of venom proteins to understand their exact 3D structure.
Advanced imaging allows scientists to map each atom in the toxin's structure.
The combination of structure and effect reveals how to design synthetic versions for medicine.
Compound A42 shows promise in targeting specific pain receptors without nerve damage.
Freeze-drying stabilizes venom compounds into solid form for safe storage.
This powder form keeps the venom stable for months while retaining all active properties.
Inside cryogenic vaults, scorpion venom is kept at -80 degrees Celsius until required for formulation.
The formulation lab converts raw venom into measurable, safe research doses.
Accuracy is vital. Even a fraction of a milligram changes pharmacological results.
Once diluted, the venom's medical journey begins. Procurement, from molecular analysis to real-world testing.
We track how each compound interacts at microscopic levels: prevalence, timing, response, and stability.
Access to venom test zones is limited to trained biosafety personnel.
Precise microdosing ensures safety while maintaining scientific accuracy.
If results remain stable, the compound could move to preclinical testing next month.
Here, science witnesses how venom's complexity could help calm chronic pain instead of causing it.
Venom compounds are coded and cataloged like pharmaceuticals, Inc., ready for regulatory review.
Every vial carries a unique serial and QR tag linking back to its extraction source.
Automation minimizes contamination risks in venom packaging. These ampules now contain stabilized venom compounds ready for research distribution. Each package must maintain cold chain integrity until arrival at medical centers.
Even in transport, the scorpion venom remains under constant monitoring for temperature and vibration.
Shipments travel across borders to research centers studying neurological and cancer treatments.
From the desert to the world, Rincon, a journey of science, precision, and hope.
Venom arrives at national biomedical centers for further formulation and collaboration.
We verify chain of custody for every batch before medical testing proceeds.
Early preclinical models help determine safety and dosage thresholds.
The compound moves closer to becoming a clinically viable pain management therapy. Venom-based peptide shows significant reduction in nerve signal overactivity.
Samples are allocated for collaboration with institutions worldwide.
From this desert lab, scorpion venom now travels to research partners across continents. A journey that begins with a sting blow and ends with hope for healing. At partner laboratories, these compounds begin their transformation into modern medicine.
From the desert's smallest creatures comes one of medicine's greatest hopes. This is the science of scorpion venom.